This is part 2 of the cell communication lectures. I wanted to come back to this practice question because I realize it's asking more than just when it says what kind of signaling molecules bind to cell surface receptors. The question means is to ask, is the signal hydrophobic or hydrophilic? So it's going to be a hydrophilic ligand that binds to cell surface receptors, whereas hydrophobic ligands tend to bind to um, intracellular receptors. Now, what we're looking at here is an ion channel linked receptor. The signaling molecule binds to the receptor and the channel protein opens. So we call these gated ion channels. And that's what happens is that the signaling molecule binds to the binding site on the channel. You can see here that it's closed. The gate is closed. And what it does is when that signaling molecule binds to the channel then opens and certain ions are allowed to pass through that channel by diffusion. Then we have receptors called G protein linked receptors. These receptors bind a ligand, such as what you see here. Here's your signaling molecule or your ligand binding to that cell surface receptor. And then that activates a membrane protein called a G protein. The activated G protein then interacts with either an ion channel or an enzyme in the membrane. So here we see the signaling molecule binding, and we see in the next step, first of all, we see this whole complex here. Sorry, my pen's not writing. Let me try this again. This whole complex here, it still isn't. It may be dead. I'm sorry. I had it. There we go. I had it screwed the wrong way, and so it wasn't working. But anyway, what we're looking at here is a molecule called GDP. Now, I know you've heard of ATP and ADP, which are adenosine triphosphate and adenosine diphosphate. Well, this is guanosine. Guanosine, I think it's spelled like this. Guanosine diphosphate, which contains an alpha unit, a beta unit, and a gamma unit. So what you see happening in this next step is the GTP replaces the GDP, and the GTP and the alpha unit remain attached to the the cell receptor, and the beta and gamma units detach. And then it says the GTP is hydrolyzed to GDP. In other words, the phosphate is removed and it becomes GDP. And that will allow some intercellular process. It will trigger some process to start. Enzyme-linked receptors. Okay, so we have a receptor, for example, tyrosine kinase is an enzyme. The receptor tyrosine kinase, when signaling molecules or ligands bind the receptors dimerize. Um, and what happens is that the tyrosine residues on the intracellular domain are phosphorylated, and that means a phosphate is added to them. They're phosphorylated, triggering a downstream cellular response. In other words, just triggering some cellular response. Whereas the G protein triggers a response when um, the beta and gamma units are removed from the GDP complex. Um, a cellular response is triggered by that. And here, a cellular response is triggered by phosphorylating tyrosine and adding a phosphate group to tyrosine. So basically, we're looking at how ligands bind to receptors, but what really happens um, whenever we say that they trigger something to happen inside the cell? Well, in a signaling pathway, second messengers, enzymes, and activated proteins interact with specific proteins, which in turn activate a chain reaction that leads to a change in the cell's environment. So if we look at the figure at the top, upon the binding of epidermal growth factor or EGF, to the epidermal growth factor receptor. So here's growth factor, here's the receptor. Two proteins associated with the receptor called GRB2 and SOS activate RS. So these two activate RAS, I'm sorry, which is a small G protein, and we know that what the G protein does is remove a phosphate from GTP, converting it to GDP. Um, then it says a protein kinase called RAF is activated by RAS-GTP. So this protein is activated by the combination of RAS and GTP. RAF phosphorylates MEK, which in turn phosphorylates ERK. That phosphate from the MEK is added to the ERK. A MAP kinase, the MAP kinase. There's a lot of three-letter words here, but it's basically just showing you that there's a chain reaction, a reaction that signals another reaction to occur. It's like triggering a pathway or a cascade of chemical reactions. So the phosphorylated ERK enters the nucleus where it triggers a cellular response. So this ERK containing the phosphate enters the nucleus. It can stimulate cell proliferation, meaning cell growth, cell migration and adhesion, angiogenesis, which is the growth of new blood vessels, and this also inhibits apoptosis. A signaling pathway, a single pathway can branch off toward different endpoints based on the interplay between two or more signaling pathways. As we saw here, we saw that this was one example, and then this was another example of how the same signaling pathway could work. Signal integration is when signals from two or more different cell surface receptors merge to activate the same response in the cell. This process can ensure that multiple external requirements are met before a cell commits to a specific response. So this would be like um, extra checkpoint. It's something that um, an extra checkpoint that's required so that the signal doesn't happen by accident. The effects of extracellular signals can also be amplified or greater by enzymatic cascades. Intracellular signaling. A major component of cell signaling cascades is the phosphorylation of molecules by enzymes known as kinases. Phosphorylation adds a phosphate group to serine, threonine, and tyrosine residues in a protein, and this changes their shapes and activates or inactivates the protein. Small molecules like nucleotides can also be phosphorylated, like adenosine is phosphorylated to make ADP and ATP, and guanosine is phosphorylated to make GDP and GTP. Okay, so phosphoserine, phosphothreonine, and phosphotyrosine are phosphorylated kinases. They are phosphorylated, excuse me, they are phosphorylated by kinases. Second messengers are small non-protein molecules that are used to transmit a signal within a cell. Some examples are calcium ions, cyclic AMP, which is adenosine monophosphate, and that is abbreviated little c, capital AMP, diacylglycerol, and inositol triphosphate, or IP3. So here's your ATP, and here's your cyclic AMP. ATP is used to produce cyclic AMP by removing two phosphate groups from it. And as you can see here, these are the two phosphate groups that are removed, and then one phosphate group is left. So the phosphate groups that are removed are here, and the phosphate group that was attached, that's your AMP, and now it's called cyclic AMP. Many pathways influence the cell by initiating gene expression. Some pathways activate enzymes that interact with DNA transcription factors. Others modify proteins and induce them to change their location in the cell. An inhibitor is a molecule that binds to a protein and prevents it from functioning or reduces its function. The MAP kinase, ERK, phosphorylates MNK1. So that phosphate group attaches to the MNK1. In turn, that phosphorylates ELF4E, so the phosphate group is added here, which is associated with messenger RNA. The messenger RNA unfolds and protein synthesis begins. So this triggers protein synthesis to begin in messenger RNA. And to this point, we've not really learned about protein synthesis, but basically it involves this. The DNA in the nucleus contains genes for your traits, but DNA has to be converted to messenger RNA, and then that messenger RNA leaves the nucleus and attaches to a ribosome and produces the protein. So basically, the job of messenger RNA is protein synthesis. And it shows you here the phosphorylation of MNK1 and then the phosphorylation of ELF4E, and that initiates protein synthesis in the messenger RNA. Cellular metabolism and growth.